Yeah, Manu, I think we'll start.
Yes. Welcome back, everyone, for the first scientific session of this conference. I now call upon Dr. Umapati to introduce the plenary and inaugural speaker.
Thank you, Manu. Good morning, good afternoon, and good evening, everyone. It is a great pleasure for me to introduce our speaker today, who is going to talk to us on hormone monitoring as an important tool to aid breeding management of wildlife species. Dr. Jalen Brown is head of the Endocrine Research Laboratory at the Smithsonian Conservation Biology Institute, and she has over 35 years of experience conducting research to increase knowledge that will lead to better management and conservation of endangered species. Using techniques to measure hormones and other biomarkers in serum, plasma, urine, feces, hair, and saliva of dozens of species, she has amassed an extensive biological database of over 150 species, demonstrating the diversity of reproductive health stress response mechanisms that exist in the natural world. Her main area of interest is elephants, where conducts collaborative research on reproduction, health, and welfare of elephants in the US, in Asian and African range countries. Dr. Brown has also called a world authority on elephant reproductive biology, and she is also in charge of elephant reproductive program at a National Zoological Park in the USA. I welcome Dr. Jalen Brown to present her work.
All right, thank you very much. I think I'm going to turn off my video and share my screen. Let's see, I need to get rid of that.
Yeah, you could see.
Do you know how to get rid of the thumbnail?
We are able to see. We are able to see your slide, yeah.
Okay, all right. Yes, well, I want to thank Dr. Umapati very much for inviting me to open this important e-conference and give myself a chance to tell you a little bit about what I've learned over the past 30 years of the importance of hormone monitoring to improve breeding and management of wildlife species, especially those under human care. I might be a little bit biased, but I happen to think that of all the techniques that have been developed for conserving wildlife over the past couple of decades, endocrinology may be one of the most important because it lets us clearly understand how factors in the captive environment directly affect physiological function, which then gives us a chance to try to change those situations and provide more ideal conditions for animals. And so that work is quite important, and it won't let me forward my, Oh, okay. Let's see. Okay, so as he said, I work for the Smithsonian Conservation Biology Institute, which is a beautiful area in Virginia, rolling forested hills. It's a conservation center of about 1,200 hectares. We have about 30 species and over 400 animals, all kind of parts of recovery programs and animals that we are studying heavily to try to figure out the best way to conserve them and create self-sustaining populations. So I was hired, as I said, in 1991 to be a research biologist, a comparative research biologist, although my first assignment was to characterize the endocrine patterns of all 36 species of non-domestic felids. So I worked pretty hard for about 10 years. I got about halfway through the species, but then also started getting sidetracked on a bunch of other species, including elephants, as Dr. Uma mentioned. They've become rather a passion of mine. But what happened when we were doing our basic research and we were developing a lot of new ways of looking at hormones in wildlife, including developing some non-invasive techniques, we started getting a lot of requests from other facilities that wanted to use these techniques to help them better manage their species. they would send samples to us to ask, is this female cycling normally, or is this female pregnant, or can you tell me if this individual is highly stressed because we just changed the exhibit? So what we ended up doing was creating the first diagnostic lab for wildlife. And we get in several thousand samples a year now, trying to help other facilities do the same sort of thing that we're doing in using these biological methods to improve management. And so then we published a lot of papers on the techniques we developed, and we started getting additional requests from people that wanted to use those techniques in their own research, students at other universities. And so we ended up setting up the first training program for teaching the next generation of scientists that want to use these endocrine techniques to monitor and study wildlife species. So we also partner with a number of universities so we can actually co-mentor graduate students and postdoctoral fellows. So as he said, we now have data on over 150 species in our laboratory. So we are a truly comparative group. And I think that's the part that's the most fascinating to me, is just how much variation there is across the different species in the types of endocrine mechanisms that you see. So this work is important because, the number of animals that are being put on these endangered species lists is only growing. And so we need better ways of conserving them. And so it's important to conserve them in the wild, of course, but one of the things that we can do, coming from a zoological facility, is that we can use captive management as a way to create insurance populations of animals before they go extinct. And the beauty of that is, in captivity, you can study species biology often in ways that are not possible in the wild. You can study the same animal for long periods of time. You can get fairly close. You can collect a variety of different sample types that you probably couldn't get if they were roaming free in the wild. And with all that information, we can do a much better job of figuring out how best to conserve them. But of course, these zoo captive studies have their own challenges, in that we're working with small geographically dispersed collections. Most zoos only have, a few individuals of each species. So you have then to rely on doing multi-institutional studies. So the limited numbers of animals for study can sometimes be a hindrance. These species primarily require non-invasive approaches. And so what we're finding is that you almost have to develop almost species-specific non-invasive approaches, depending upon what your animal is and what your question is. And then finding that breeding problems are often very species-specific. So there are a lot of causes of poor reproduction among wildlife species, especially in captivity. We're seeing, that loss of genetic variability, this was known probably 40 or 50 years ago, has a negative impact on reproduction, and so think about the cheetah, that had a population bottleneck about 16,000 years ago, and they have very low genetic variability, but also have difficulties with health and reproduction. If captive facilities don't have the right environmental factors, or they're using poor husbandry, the important part is that you should be trying to meet their natural history needs. So putting a polar bear in a desert exhibit is probably not good for its welfare. Bird species need to be in enclosures big enough so they can fly. If animals are rather aquatic or like to spend a lot of time in the water, you really need to be providing them with a pool so that they can do that. So then that brings out their natural behaviors, and you're going to get much better reproduction. Poor nutrition is something that, it's surprising how little we know about the nutritional needs of so many of our species. And very few institutions actually have their own professional nutritionist on site. So a lot more work needs to go into that, and nutrition can certainly have an impact on reproduction. And then even if you, have ideal, conditions, and you get males and females and put them together, they may not breed because they are socially incompatible, or there's not enough mate choice availabilities. And so really kind of getting socially compatible groups that are willing to breed can be more difficult than you think. And then of course, the overall concern about whether these animals are in general stressed due to their environment. So when animals don't breed naturally, people have developed a variety of reproductive technologies to try to preserve species that might not breed on their own. So semen collection and cryopreservation, gamete rescue, taking these frozen tissues and keeping them in a genome resource bank so that you can then use them later for assisted reproductive techniques like artificial insemination, in vitro fertilization, and embryo transfer. And then, of course, there's endocrinology, which can map the different cycle phases of different animals. And it's also key to the development of these assisted reproductive techniques, because a lot of them do rely on hormonal manipulation in order to get them to work. So endocrine monitoring during the development of these techniques is key. But it's not just about producing offspring. The idea shouldn't just be to produce as many offspring as you can. They have to be quality offspring. They have to be properly genetically managed so that they will thrive in a captive situation and be genetically healthy if they are going to be used for reintroduction purposes. So the goal is to maintain 90% of genetic diversity for the next 100 years. And this is something that zoos do a really good job of, tracking pedigrees and genetic relatedness, and then helping facilities decide who should be breeding with whom. And they will do that using, we always try to do natural breeding if at all possible, but then we now have a toolbox that allows us to do assisted breeding if animals can't or won't do it naturally. So I kind of like the analogy of a house. When you build a house, maybe one of the most important things that you build is actually the foundation. So you want to build the foundation first. It's got to be really strong because without that, the house is likely to collapse over the years. And so I kind of look at endocrinology as being the foundation upon which we build everything else in terms of wildlife species management. And so through endocrinology, we have a much better understanding of how these different factors are affected by our captive environment. And of course, breeding, whether natural or using assisted reproductive techniques. So I always kind of look at, and endocrinology is often the science that you don't see very much of. We're not producing our own babies or, doing things that are very exciting, but it's right there. You don't see it very often, but it's very important. So in our lab, our primary goal, first and foremost, is whenever we get a new species, a request to study a new species, the first step is to just understand that species biology. So we conduct basic research really just to understand what their reproductive patterns are. When does reproduction start? When does it stop? Are they affected by season? And then what other factors in their environment might impact reproduction and welfare. And then once we have that normal data for an individual or for an individual species, we can then use that information and compare it when we find problems in reproduction in some places. So we can assess fertility status and determine if there is a problem. And if there is a problem, we can very often identify what that is and treat it. And of course, welfare is always very key to what we evaluate here, because without good welfare, you're not going to get good reproduction. So there are a lot of hormones obviously involved in the reproductive processes, as well as health and well-being. So over the past 30 years, we've developed a lot of different assay systems to measure a variety of hormones, not just your typical gonadal steroids, that we've been able to apply to sort of understand the interactions among different endocrine systems and how that directly impacts reproduction. But I'm in animal science. That was my major. And so I studied dairy cattle, sheep, and some other animal model systems. But we were able to do all of our hormonal analyses using blood samples. So when I came to the National Zoo to start working on wildlife, all the assays that I brought were based on being able to collect blood samples, which for some animals is possible, but for the majority is not. So the minute I came to working for the SCBI, the first step was to really start developing a variety of different non-invasive approaches for animals. which actually turned out to be easier than you would think, because the endocrine products that are secreted into the blood come out of the body in various ways and forms. So they can be directly secreted into saliva, milk, and hair, and you can measure hormones there. They might go to the kidney, where they are conjugated to make them water soluble, so they'll come out in the urine. Or they can go to the liver, where they are usually more heavily metabolized, so they can leave the body through the bile into the gut and then out into the feces. And so it's really important to know for your particular hormone how it's being metabolized and excreted, so that you can develop appropriate assay systems to monitor them. And so, and also keep in mind that you can't necessarily measure everything everywhere. You certainly can in the blood, because that's where all of the hormones originate. But you need to keep in mind that most of the hormones, especially the steroids coming out in urine, are going to be conjugated to make them water soluble. Some species will secrete or excrete some proteins, but not all. And so we've run across that with some of our species, where you get really good recovery of protein hormones in urine, but with another species, you can't find them at all. Saliva and milk, those are basically free steroids, so you can use almost any assay that you want. Proteins are also found in saliva and milk, and we're starting to measure immunoglobulins as a measure of health and well-being that often are in these matrices. Keep in mind that the fecals, the liver heavily metabolizes steroids, and so you have to find more group-specific assays to measure those. But we've started using feces for looking at metabolic function and then health function with the IGs. And then a kind of a newer technique is the deposition of steroids across a keratinized sample. So free steroids being able to be measured in hair feathers and baleen. So the beauty of the non-invasive monitoring, obviously, is it doesn't disturb the animal, and you can collect samples over long periods of time with the animal not even knowing it's being monitored. Urine and fecals are the most common. And those two represent a pooled sample over time, which definitely has its advantages, because a lot of hormones are in the blood, secreted in the blood in a pulsatile manner. And so if you're just going to go in and collect one sample, you don't know whether you're going to be collecting it at a peak or at a nadir, because you're not doing longitudinal sampling. So that can maybe help you misinterpret the data that you have. And other hormones are secreted in a diurnal fashion, which means it's very important for you to collect the samples maybe at the same time of day, every day. But with urine and feces, it actually pools samples over time, depending upon how often an animal urinates or defecates. And so it almost creates like an average hormone concentration over that time period, which, unless you're really looking for those dynamic changes, this actually provides a nice way of looking at overall hormone levels without being worried about all the dynamic spurious changes that occur. So when you've got a new species and you've got, you're being asked to help the zoos do a better job of breeding them, you have to ask what the research question is first. What is it that I need to understand? What do I need to look at? This is where you would say, is it okay for me to use kind of overall averaged concentrations over time, in which case urine and feces would work quite well? Or are you interested in those dynamic surges in response to short-term acute changes, in which case you might have to use blood? And then you have to determine the major route of excretion, where they're coming out, keeping in mind, and then keeping in mind how you can measure those steroids and whether they're free or conjugated or heavily metabolized. Protein hormones are another thing to consider because of the amino acid structures. They will differ across species. So whereas a testosterone in an elephant is the same testosterone you would find in a cat, with protein hormones, they're not going to be the same. And so we know that we can't measure LH, luteinizing hormone, using a human kit in elephant, but we can do it using bovine reagents. So it takes a little more time to figure out how to measure protein hormones. And so the key really at the end of the day is you need to make sure you're using the right assay to measure each hormone. So in the early days, when we really didn't know very much about hormone excretion, we focused a lot on the steroids. And one way to figure out where they're being excreted and how they're being metabolized is to do a radiolabel infusion study, wherein you inject small amounts of C14 labeled steroid, and then you collect all the urine and feces for several days and measure the radioactivity, which tells you then whether it's coming out in urine or feces or both. And then you can take those radioactive samples and you can subject them to further purification steps and actually identify specific metabolites so that you know what you're looking for in each of those compartments. And of course, nothing's easy because there's a lot of species variation in terms of how animals metabolize and excrete hormones, of course. So this is just an example of some early studies that were done in the 90s, looking at estradiol and progesterone. So if you look at the top on the domestic cat, you can see that, and we were kind of surprised that almost all of the steroids were coming out in the feces. There was very little coming out in the urine, which would explain why some of the earlier studies trying to track cycles with urine were not successful in cats. And then looking at wild dogs, they have urine and feces contain these steroids and in about the same proportions for both. So that makes it very easy. You can pick whichever substance you want, and they seem to act the same. But then if you look at the Sumatran rhino, there's hormones come out in both urine and feces. So again, you could use both if you wanted to, but if you want to do progesterone, it barely comes out in the urine. So you have to use fecal samples if you want to look at luteal activity. And then just the opposite is true in the elephant, where there's very little estradiol that comes out in feces. And so really fecal monitoring has not been useful at all. You have to use urine, which can sometimes be trickier to collect. But if you're doing progestogens, luckily you can use either sample type to monitor luteal function. And that's mostly what we do in terms of tracking elephant cycles. So there's just a lot of variability across species in terms of how they excrete things, and also differences between the actual hormones and how they are being excreted. The other thing you need to keep in mind is the hormone lag time. So we consider blood real time. And then the other hormones will start coming out over a matter of minutes to hours in something like saliva. It could be hours to maybe a day for urine, again, depending upon how often an animal urinates. Feces can take several days even for larger animals to defecate and get you a sample. We can look at hair, which accumulates over a matter of months, and so can give you real long-term kind of chronic changes in these hormones. And then more recently, people like Kathleen Hunt have developed a way to sample a baleen plate across its plate, and she can actually get years of longitudinal data from these animals, which is really cool. So hormones can tell you a lot. And again, we're always trying to get back to the basics in terms of understanding the basic biology of each species that we're working with, and then using that information in a very applied or practical way. So if we need to develop assisted reproductive techniques, we will know how to do that because we know what the hormone profiles look like. We can identify reproductive problems like ovarian acyclicity in elephants, for example, or the discovery that cheetahs can actually suppress each other's reproduction if they're housed together. And looking at glucocorticoids and other welfare biomarkers, it's very important to consider stress and welfare when you're trying to raise animals under human care. So there are just a lot of different reproductive mechanisms that we see across these various species. And this is another piece of information that you're going to want to know when you start working with a new species. So you need to understand what kind of ovulation they have. Is it induced or spontaneous? Do these animals show delayed implantation or pseudopregnancy? Are you able to actually diagnose pregnancy? What are their estrous cycles like? Do they have one? Do they have multiple? Or are they seasonal? And the interesting thing is that even with taxa among fairly closely related species, you might see a variety of these different mechanisms within that. So, the tiger isn't necessarily the same as a margay in terms of how their reproductive mechanisms play out. So these are all very important questions to ask. And so very just simply, the two types of ovulation are spontaneous at the top and induced at the bottom. With spontaneous, everything just goes, there's no outward force to create these cycles. You have a wave of follicular development that stimulates an LH surge. The follicle ovulates and then produces progesterone during the luteal phase, and then it just spontaneously starts all over again. Whereas with induced ovulators, that waves of follicular activity or just a lot of follicular activity is then triggered to have an LH surge only after they've been mated. And with some species, they have to be mated multiple times in order to induce an LH surge. And you can kind of see the list of different species that we've looked at that have these different ovulatory patterns. But interesting, when we looked at, felids are in their classic textbook induced ovulators, but we now have a lot of examples, I would say, of the, 20 species of cats that I looked at, half of them actually would exhibit spontaneous ovulation, even if they were not in the presence of a male or even in the presence of another female. So you can see this cat has waves of estrogen activity in green, followed by what's ovulation and progesterone production. And she did this 3 times in this period, but then all of a sudden towards the end, she's not ovulating at all now. So now she looks like a classic induced ovulator. So we even have species that seem to be able to do both. So I think that just adds a little bit of flexibility to their reproduction, that they are more able to respond to outside influences, or if a male kind of wanders through at times, they're better able to respond and breed and perhaps conceive. But that was an unexpected finding. The giant panda is a really interesting one. They exhibit delayed implantation. They're also monestrous. They only have one estrous cycle a year. And so they're only fertile for about 36 hours over a year. And when they ovulate, they undergo a change in the progestrogens during the primary phase. Now, if she's pregnant, the embryo just floats around in the uterus for variable lengths of time. It can be 50 to up to 150 days. And then there is a trigger of some kind. It could be photoperiod, it could be nutrition. We don't really know exactly. But then that embryo will implant, and then you start seeing a large increase in the production of progestogens. And then the fetus starts growing over a much shorter, very consistent time period. So their secondary phases last about 40 to 50 days before they give birth. And when they do give birth, they're extremely small. We say they're about the size of a stick of butter, and they're completely helpless. So, our joke is we kind of wonder how the panda itself hasn't gone extinct, because it has a very complicated breeding mechanism. And then the other thing that sort of stymies us scientists is that they also exhibit pseudopregnancy. So if they don't get pregnant, their hormone patterns look the same as if they are pregnant. And they also show very strong behaviors, whether they are pregnant or not. So it's like they go through all of the actions as though they were pregnant, even if they're not. So diagnosing pregnancy in the panda is particularly difficult. But we do use hormone monitoring throughout to time breeding, and also at least get a sense of whether she's ovulated or not. And then we look at seasonality, pretty self-explanatory, but there are a number of species that just cycle all year round. They're not influenced by photoperiod or other environmental cues. Some are just seasonally polyestrous. And then, like the giant panda, we have seasonal monestrous that sometimes occurs. So we've looked at a lot of different species that are exhibiting these different types of estrous patterns. And so looking at the cats, here's a nice example of a non-seasonal. And so this would be a induced ovulating profile, because we don't have any progesterone. She's not ovulating at all. And so we have a variety of other cat species that fit that model of being non-seasonal. And then we have a number of cats that have a slight anestrus period. So clouded leopard during the late summer and early fall, usually tied to a bit to photoperiod. And then a number of other species that we have found that have similar profiles. The most seasonal felid that we have identified today is the Pallas cat. They live in very north, cold, harsh climates in like Siberia and Mongolia. So it makes sense that the female would have a very, very tight breeding season. And you can see her estrogens just in general kind of creep up as the day lengths get longer. If you look at the male, you can see his androgens are starting to come up before the female, and that's probably to make sure spermatogenesis is fully complete by the time the female enters estrus. And so he's ready to breed with her when she is at her best. So if you look at this profile, this was in one zoo, and then this was the profile from another zoo. And you can see there was this kind of premature increase in October. And then it went away. And when the real breeding season came, they were shifted. And so the breeding season itself overall was a bit later, and the male did not reach his peak fertility until after the female did hers. So these Pallas cats at this particular zoo were not breeding well. They hadn't been breeding well for years and years, and they did not know why. So we went back and said, what's happening during this November period? And they said, oh my gosh, we have a festival of lights at the zoo where we turn all of the lights on at night so people can come in and kind of enjoy our fall festivals and things. And so it was just enough light to throw these cats into a premature breeding season. And then when the lights went off, they kind of went back to normal, but then they were way asynchronized in order to come back and have a good breeding season during the right time of the year. So they moved the cats away from this area and have been producing Pallas cats ever since. So this is a kind of a fun example of how endocrinology can actually give you some very serendipitous insight into what's going on with your animals. And zoos are now paying very close attention to what they're doing around the very seasonal animals. Okay, so we worked a lot with cheetahs at the Smithsonian. And as I said, they have low genetic diversity, which seems to be related to poor reproduction and health issues. And when I first joined the zoo, the only way they had really of looking at reproduction was to do laparoscopic evaluations of the ovary. And they constantly found these inactive ovaries. So this was the first species that I developed, the non-invasive fecal monitoring. And yes, in fact, most of them weren't cycling very, very normal. But then the more animals that we started to evaluate, the more we got a sense of, it may not just be all or none. We had seen groups of cheetahs that seemed to suppress each other. Cheetahs are not an aggressive species in particular. And so a lot of zoos, because they make a good exhibit, will keep them together. So we actually did an experiment where we put pairs of cheetahs together for 6 months, and then we collected daily fecal samples. And in all of our pairs, you can see a decrease in follicular activity while they're together. And then when we separated them, all of these cheetahs started to cycle. So there was, even though they weren't overtly fighting, there was some repression going on. And so today, at least at our place, if we want to breed our cheetahs, we keep them separate. And so that was another kind of nice accidental discovery about just using hormone monitoring to characterize a particular species. And then lions was another interesting problem that we worked on. So historically, they breed really well in captivity. And in the United States, they were breeding so well that there was a period where they just had too many and they didn't have places to put them. So the decision was made to undergo a breeding moratorium and just not breed them. So they did that for about 8 years. And then when they lifted the breeding moratorium, none of the cats were breeding successfully. And so nobody knew, what the problem was. So we did a hormonal evaluation of the captive population. This was with a graduate student of ours, Sarah Putman, to see just what was happening with these lions. And a lot of them were, just sort of having baseline hormone concentrations. The estrogens aren't doing much. The progestogens are definitely a baseline. She's not ovulating, although she is being bred. There just isn't anything happening physiologically with the hormonal axes. So that was one type of abnormality we saw. And then there were a group of cats that actually were breeding and ovulating and going through these non-pregnant luteal phases, like here, but they weren't actually getting pregnant. So these repeat breeders, were another concern that we didn't quite understand. And then when we started looking at how the animals were being managed during the moratorium, we found out that a lot of the zoos were putting these cats on contraceptives to shut down pituitary ovarian function. And they would use either synthetic progestogens as an implant, or they would implant GnRH analogs, super active analogs. And these implants were supposed to only be functional for, a year or two, perhaps. And then many of them could be physically removed. But what we were finding is that even when they were removed, these cats weren't coming back to cycling for, 3, 4, 5, some of them 7 or 8 years after they had been put on contraceptives. So the contraceptives had wiped out the population for, a good, close to a good decade. So we're very careful now about how we use contraceptive in these cats and not putting them on for very long periods of time. And then, yes, my favorite species, I have to admit, are elephants. These are the 4 girls that were at the National Zoo when I first joined. In fact, I even started working on them before I was hired by the Smithsonian. The curator had asked, I was working up the street in another university, and he called and asked if I could tell whether the youngest female was cycling or not. She was 12 years old. We didn't know much at all about the biology of elephants. We didn't know when they came into puberty, but they were interested in breeding her. So we started bleeding her a couple of times a week. We were going to do it for maybe a year and a half, just kind of figure out if she's cycling. And in fact, you can see that she is cycling very normally. And so the decision was to send her up to Syracuse, New York for breeding. And so they were going to stop the hormone monitoring. And I said, no, let's keep going. Let's see what her pregnancy looks like. So we don't know that much about pregnancy in elephants. And so they brought her, she was pregnant, they brought her home. And then I said, well, let's keep monitoring her after she gives birth, because we want to know something about the lactational anestrus period. And then what does it look like when they start cycling again? And, every time we would finish something, I would have another question or two. And so whereas this was only supposed to be a very short-term project, it obviously ended up being a 30-year career. So yes, my ultimate goal for elephants is just to understand their biology first and foremost, but then use that information to try to optimize reproduction, health, and welfare. And I initially started all of my work in the United States, working with zoo elephants, but I have since now moved to doing a lot of my work in Asia, trying to kind of replicate the same things that we do in the US, to do them in Asia, again, to try to improve breeding management and welfare of captive elephants in other places around the world. And so it was really the elephant work that we did and all the papers that we published that got us into doing a diagnostic lab. And so we still work with most of the zoos in the country that have elephants will send samples to us. And we do a lot of pregnancy diagnosis, for example. So, and Steve Paris is my technician extraordinaire. So with all these elephants that we had to evaluate, we were able to, kind of do little research projects with them. And so fairly early on, we developed the model of the elephant ovarian cycle. They have the longest cycle of any mammal. It's between 3.5 and 4 months long. They have about an 8 to 10 week luteal phase. And then their follicular phase is also very long. It's between 4 and 6 weeks long. And during that follicular phase, they have 2 waves of follicles that grow. And at the end of each wave, there is an LH surge, but the first LH surge does not induce ovulation. Rather, the follicles that were there luteinize, and then they become active after the second LH surge to presumably produce more luteal tissue that produces more progesterone, so that if the elephant does get pregnant, they are better able to carry it. But this is a very unusual profile. We don't see this double LH surge in other species. So this was a really fascinating discovery. And we were looking at other hormones to try to figure out, why you have these two waves and why only one ovulates. And there are enough differences in the hormonal milieu between the two, especially when you look at FSH and inhibin. So the two hormones are inversely related with each other. And you can see that during the first surge, the FSH is fairly high, and then it starts to decline about the time the inhibin starts going up. And so by the time you get to the second surge, you've got the decrease in FSH, which is one of the factors that allows dominant follicle selection. You have an increase in inhibin, which also indicates that those follicles are becoming more mature and being able to produce inhibin. So it seems that that is the milieu that is important for the maturation of the follicle that allows dominant follicle selection to occur. So this took quite a few years to figure out. And so, this is just kind of a cool profile from an elephant at the Oregon Zoo, and it shows the initiation of cyclicity in a very young elephant. And we had always assumed that elephants were reaching puberty, probably 8, 9, 10 years old, something like that. But we're finding in our zoos, especially Asian elephants, that they can start much, much earlier. They can be 4 or 5 years old. And so zoos have to be really careful. Hormone monitoring is essential if you've got bulls and females hanging around together, because accidental pregnancies of very young cows have occurred in the past, and you don't want that to happen. So then the gestation is about 20 to 22 months, which is also the longest of any mammal. And then you can kind of see this nice lactational and estrus period at the end. And then when they do start cycling again, it looks exactly the same as if it was just a normal cycle. One fun thing we can do if we have a pregnant elephant is we can tell whether she's carrying a boy or a girl by measuring testosterone in the mom's blood. And so after about a year, the maternal testosterone starts to increase, and that is a boy. And then if it stays fairly flat, we know it's a girl. Oh, but that only happens in Asian elephants. And I don't really understand why. There's obviously enough of a difference in the uterine physiology that for whatever reason, we don't see these clear patterns in the African species. The other thing we can do is we can use hormones to tell when an elephant is going to give birth. Because she usually gives birth between 3 and 5 days after the progesterones drop to baseline, as opposed to other species where when the progesterone drops, that's usually when birth occurs. So our service lab does this all the time. We monitor progesterone in daily blood samples towards the end of gestation, so we can call up the zoo and tell them to get ready. And so they really appreciate the fact that we can do this for them. And so how to breed an elephant, it's a big challenge given their big size. We prefer natural mating if at all, but if that doesn't work, there's artificial insemination. But each of these methods has their own challenges. With natural mating, oftentimes it's an opportunity issue where a zoo doesn't have males and females together, or a zoo might have a male and female, but they're not compatible. Or the bull, they seem like they like each other, but the bull just doesn't know what to do. And so he doesn't have the ability to breed, which can be really frustrating. So in those cases, if the genetics are really important, we'll start to rely on artificial insemination technique, which was actually something that was started at our zoo, the National Zoo, with Thomas Hildebrand and Frank Goritz. So the challenges with AI is you need to get a good, hardy, fresh semen sample with good motility. You need to be able to time it properly because they're only fertile. The female's only fertile for maybe 36 hours every 3 or 4 months. And then you need to place it in the right place because the reproductive tract is very long. It's about 3 meters long. And you need to get it into the cervix or the uterus in order for it to be successful. So for collecting sperm, we use a rectal massage technique that was actually discovered by Dennis Schmidt. Some bulls do really well with this technique and can produce really good semen samples. Others, unfortunately, don't, even though they are very fertile bulls. So if you get a bad collection, it doesn't necessarily mean the bull has a fertility problem. We think that some of the problems may just be the wrong mix of accessory sex fluids that give proper, modal stimulation to these sperms. So this is a technique that still needs some additional work to try to get it to work. And then the timing is probably the easiest part. And that's my job, is to time the AI, because we come back now to this double LH surge, and they occur 3 weeks apart pretty consistently between both Asian and African elephants. And so we do daily blood collection until we identify the first surge. And then we know that we tell the teams that, they should come and do an AI about 3 weeks later. And so that's been working brilliantly. And then, as I said, she's got a really long reproductive tract. So the way they place the semen is they use a balloon catheter first. You might be able to see that here. And they insert that in the vestibule first. And then they thread the endoscope, which has the semen catheter, and they'll thread that through the balloon catheter so that it can kind of get up into the cervix. And they try to get it into the uterus, but the proximal cervix works as well. And so this has been a very valuable breeding tool for us in the United States, and a lot of calves are produced by AI. And so, yeah, the advantage of long-term hormone monitoring, especially species like elephants, where you can bleed them fairly consistently, is you can learn an awful lot about their reproductive patterns. These are 2 different pregnancies. One was natural, one was AI, the left was a female, the middle was a male, and just sort of get a sense of what their overall biology is. So the cycling elephant is because somebody told me every time I talk about cycling elephants, they think of one riding a bicycle. So now every time I talk about cycling, I think of a bicycle. But this is another one of our elephants who is the oldest one in our group, Ampika. And as you can see, she's also cycling very, very normally for literally decades. And so in her case, as she was getting older, into her 60s, she started developing reproductive tract pathologies, which we think is the result of these females that never get pregnant, and that are constantly exposed to steroids, especially estrogens, that they start developing reproductive tract pathologies like tumors and cysts. And hers were getting so bad that it was causing bleeding. She was anemic and she almost died. So she was the first one that we did a little experiment with a GnRH vaccine to shut down the pituitary ovarian axis. And it worked brilliantly. She stopped cycling. And the tumors and cysts went away. Her uterus involuted, and she actually never did resume cycling after this. And she lived another 12 years after we did this technique. And I really think that we expanded her life because we were able to, first off, catch what the problem was and then fix it. And we wouldn't have known that if we hadn't been doing hormone monitoring. And so here we go. The more we learn, the more problems that we identify. And so we've spent a lot of time looking at reproductive cycle problems in elephants. We have found over the years that about 15 to 20% of Asian elephants don't cycle at all. We call them flatliners. These are mostly older females that are probably not necessarily needed for the breeding pool, so we don't worry about them quite as much. But about 40 to 50% of Africans also don't cycle, and a lot of those are in the reproductive aged category. So we're very, very worried about that. And so we've spent literally 15 years looking at everything we could think of that was related to fertility from an endocrine standpoint. And, we didn't see anything related to, stress using cortisol as a proxy. My PhD was in cystic ovarian disease in dairy cattle, but I didn't see anything like that in association with flatlining in our elephants. And so we looked at, they weren't hyperandrogenic. Metabolic activity seemed pretty normal. We injected GnRH to look at pituitary responses of LH and FSH, and those were normal. And so we wondered for a while whether maybe they were going through a premature menopause. So we looked at anti-Mullerian hormone, and actually that was fairly normal too. So there's got to be something else going on with this that is maybe not directly endocrine related, with the exception of the African elephant that exhibits high rates of hyperprolactinemia, which is a whole nother thing that we've tried to understand. It's found in about 60% of the acyclic African elephants. So a good number of, if an African elephant isn't cycling, she has a pretty good chance that she's hyperprolactinemic as well. This is a cause of infertility in women and livestock, and it's often related to a pituitary tumor, although we don't exactly know what it is that causes that pituitary tumor in the first place. But hyperprolactinemia is often linked to stress in women. So again, we looked at cortisol as a proxy for stress, but quite frankly, we just didn't see any differences between our cycling and non-cycling girls or the non-cycling hyperprolactinemic elephants. So if it is a stress response, it's going through a different mechanism than the adrenal gland. But the interesting thing we found with prolactin is with temperament is so the high prolactin elephants are more likely to be popular caring as rated by their keepers. And so it looks like these hyperprolactinemic elephants may be acting as peacekeepers of the herd because most of our captive herds are not related individuals. And so there could be a lot of, social strife among animals if they don't all get along very well. So we're thinking that some of these elephants are stepping up and they're kind of stopping cycling. And at the same time, their prolactin kicks in order to maintain social bonds when they're housed with a group of unrelated females. And so the big question is, well, should we be curing this problem? Because if we get rid of the peacekeeper of the herd, what's that going to do to social stability? So we haven't really approached that to any great degree. And whereas the low prolactin elephants seem to be more fearful and apprehensive. So the low prolactin, which these are non-cycling elephants that have non-cycling prolactin, they may be less able to cope with social demands, which is something we've seen in primates. So these are an interesting sets of experiments that suggest that prolactin actually may play a really important role in supporting normal social relationships with elephants. And that I think elephant sociality is probably the most important management factor that we need to address when it comes to health and welfare and reproduction. We haven't found any evidence of ovarian cyclicity in the wild, and we've done that through monitoring fecal progestogens in wild elephants and also looking at ovarian structures on samples that were collected during culls in South Africa. So that just brings us back to wondering whether is it strictly management oriented? And if we had better management conditions, would that support good welfare and therefore have a positive effect on ovarian activity? So this is when we designed about 10 years ago, this big epidemiological study in the US, and we had nearly all of the elephants that are in our AZA accredited zoos participate, where we looked at a variety of environmental inputs, environmental and demographic inputs that are shown in the circles. So their social makeup, the exhibit, complexity, how much exercise they get, their social group composition, all of that. And then we modeled them against welfare outcomes shown in the squares. And so, of course, I was particularly interested in the reproductive part of this, but the others as well, of course. And so my lab was responsible for measuring hormones in blood and fecal samples that were collected, looking at reproductive health, other biomarkers of metabolic function. We had over 6,000 samples each of blood and fecals. And in many of those samples, we were measuring 12 hormones. So it was a huge, huge study. And I'll only be able to present maybe like 1% of it. But here were some of the results that we got from looking at factors related to ovarian cyclicity. So this was just looking at the progesterone profiles and were they cycling or not. And so we met our hypothesis in that we felt the social factors were probably one of the most important in terms of whether a female had normal reproduction or not. So being kept in a mixed sex herd, having what we called good social experiences with elephants, being able to interact with other elephants in a positive way, it increased the chance that a female would have normal cycles. Experience of pregnancy also increased the chance. And we call this use it or lose it. If you don't breed an animal on a regular basis, we do find a lot of problems later on if we try to get them pregnant again. whereas social isolation. So when we have elephants that don't get along very well, they're often isolated. And even if they can interact through a howdy gait, we do find that being apart from other elephants can decrease the chance that she'll cycle normally. So we conclude that a multi-generational herd with calves and bulls and spending more time in socially compatible groups is what is really good for both reproduction and welfare. A number of management factors were related to whether a female cycled or not. A lot to do with the complexity and the diversity within the environment itself. So in terms of how many items that they had to play and interact with, and also how the food was presented. So, you know, did they have to search for their food? Did it cause them to explore their environment, which all seem to be very important for, by promoting normal behaviors, it seems to enhance physiological function. We have had some evidence that high body condition scores and just being overweight may correlate with lower fertility. This is primarily in African elephants, not Asian elephants. But in our population of the epidemiological study, 3/4 of the elephants were scored on a score of 1 to 5. They were either overweight or very overweight. So we do have an overweight problem with our captive zoo elephants. That is one of the things that, of management things that is being changed right now based on the results that we got. And then we looked at glucocorticoids in terms of, you know, it doesn't seem to have a real strong relationship with ovarian cyclicity per se, but we wanted to look at it in terms of the other welfare outcomes. And so, and it makes perfect sense that being kept alone, having poor joint health, spending time on hard surfaces, which was also related to poor joint health, and having low enrichment diversity were also all associated with having higher fecal glucocorticoids. But then having positive keeper contact, you know, elephants are very social, including with their humans, and getting a lot of exercise was associated with lower concentrations of the glucocorticoids. But then you can't overinterpret glucocorticoids, because it's not always straightforward that high glucocorticoids correlates with stress and that low correlates with good welfare, because there are situations where elevated glucocorticoids are associated with normal physiological function. So like the pregnancy, pregnancy, parturition, and having the presence of calves can be associated with higher glucocorticoids as these females adapt to being a mother. So it's just an example of how you need to be very careful in how you interpret corticoids. So what we found overall, you know, just looking at how these factors affected ovarian function, we also found that they, you know, really seem to correlate well with just welfare in general. And so the take-home message was that it's not necessarily the total amount of exhibit space that is important. It is actually much less of a factor. Rather, it's more important to have a complex environment with things that elephants can do and to stimulate their normal exploratory behavior, and then to keep them with socialized elephants so that they can be reproductively active. And so this was a little bit of a surprise. I think everybody thought the size was going to be the biggest factor important for elephants, but that's not the case. You really want to give them a very challenging environment. And so I would say with all of the work that we've done over these 30 years, I would say that probably 80 to 90% of the reproductive problems that we are seeing in captive collections is probably because they're not being kept in the right environment. We're not giving them an environment that sort of matches their natural history. And so if we could do that, I think we would be in much better shape. You know, it's fairly easy to take good care of an animal, although I suppose not everybody does that either. But, you know, just giving them good shelter, clean water, is it sanitary? Do they have good veterinary care? You know, is it a safe environment? You know, those should be the easiest things to do. But what you really need for good welfare is you need to be meeting not just their physical and physiological needs, but their psychological needs as well. And if we can do that over the long term, I think we'd have a much better chance of producing more self-sustaining populations. So with that in mind, I'm going to say goodbye and just introduce you to a bunch of people that work in my lab. These are great people. Some of them I've worked with for 20 years as well. So we have a very kind of tight-knit group that has been working together for a very long time. And I really appreciate all the hard work that they've done with me over the years. And so I thank you very much for your attention. And I hope there's some time for some questions, perhaps. Should I stop sharing?
Yeah, thank you. Thank you very much for wonderful and extensive work on elephants and other species with reference to endocrine monitoring. In fact, we are all just grown with seeing your research for a period of time. When we saw your first paper in 1994, and it was really that is it possible to someone examine hormone from fecal samples? That was a question. Still people ask, is it hormone can be estimated from fecal sample? That question you answered 25 years ago. So thank you so much for very extensive, elaborative discussion about this endocrine monitoring. We have a few questions and a few people have raised their hand. I can read some of them. The first question from Dr. Karthikeyan Vasudevan. He asks, what are the ethical considerations for ART intervention in captive wildlife?
I'm going to actually, are you reading the chats?
Yeah, ethical consideration for ART intervention in captive wildlife. What the ethical consideration?
Oh, ethical considerations for ART intervention.
Yeah.
Well, that's a hard one to ask. I mean, I don't think it necessarily has to be an ethical consideration if it's done for the right purpose and if it's done in the right way. You know, there are very safe ways, obviously, of doing assisted reproductive technology. And if I always kind of tend to look more at the big picture, and if our goal is to make sure that this animal does not go extinct, then I think assisted reproductive technologies are really, really important, and all the work that we've done on species that had gone extinct in the wild and now have been reintroduced. I think that makes it, probably the most important part.
Okay, thank you. I think the panelists can directly ask question. Can I ask Edward to ask directly your question? Are you there?
Do you want me to read some of these questions?
Yeah. So he was asking, so many species become endangered, that so many antibodies are required. Is there anything possible group-specific antibodies can be used? And is there anything we need to do every time new antibodies for new species? What can be done? That's what his question, basically.
Yeah, that's a very good question. And it's something that we face every time we have a new species. And I think you might hear a little bit more. I didn't delve into it too deeply in terms of the types of, specific versus broad spectrum antibodies, but that is certainly one of the major questions about using the right assay. So honestly, when we start with a new species, we literally go through the freezer and pull out a bunch of antibodies and start looking at all of them to see, which one gives us the best biological, information. So, because you want an antibody, if a steroid's been heavily metabolized and it may not be recognized by an antibody against the native hormone, if it's progesterone and everything that's coming out is not progesterone, a progesterone assay might not measure it. So you really have to go through and try to pick antibodies that recognize other parts of the molecule that maybe are less specific. And I think that's the biggest challenge with, and for me, when I felt like I did my first 10 years doing nothing but assay validation, trying to figure out how should we extract, what's the best way to extract, what's the best antibody to use, and just, testing all of them and seeing which one works. Now that we've got a pretty large body of information, we have a pretty good sense that maybe this broad spectrum antibody seems to work really well with ungulate species. This broad spectrum antibody seems to work really, really well with carnivores. And so we've sort of laid the groundwork where we but you still have to test it, you still have to validate it. So we have more information now that can lead us in the right direction, but you still have to validate it.
Okay, thank you. Karthik, you asked another question. Can you ask once again yourself? Unmute and speak.
Sorry?
Karthik, my colleague is asking me a question. Yeah.
Dr. Brown, can you hear me?
I can.
Wonderful talk. I really enjoyed listening to your talk. And I think your last slide was just, it just captivated my attention. It just summarized everything that you spoke about. My question is about the species that have long interbirth interval. What are the roles of the hormones in neonatal care and success in parenting? Are there any specific roles of hormones in this?
Hormones during neonatal?
Specifically in species which have long interbirth intervals.
Oh,
what is the role of the hormones? Is there a monitoring of hormones for neonatal care and parental care? Success of parental care.
Well, I mean, hormones that are involved with natal care, we've done some work with things like oxytocin and prolactin can also be, helpful for, seeing whether a female is expressing normal physiology or physiological, changes after the birth of an offspring. The length of the inner birth interval, has a lot to do with, their nutrition, for example, are they healthy individuals? It has a lot to do with the lactational anestrus period, has a lot to do with how well the offspring is nursing, that sort of thing. So I'm not sure I'm answering your question specifically, but we have done a fair amount of work looking at that postnatal period and what's happening hormonally, with a few changes in oxytocin, a little bit of work with prolactin, and and then right before birth, there's a lot going on with cortisol, for example, as it kind of prepares the animal for birth. And then the cortisol in some animals anyway, seems to stay pretty high during that lactation period, which is probably just, the stimulation of having a calf constantly to constantly keep track of. I don't know if that answered your question.
Yeah, that's nice. Yeah, thank you.
There are a few questions. One question from Sanjida. What decides such a seasonal pattern in breeding? For instance, how a light or other abiotic factors would fluctuate the endocrine pathway? Is it conserved phenomenon or this is plasticity of any sort?
Okay, is that a written question?
Yeah. What decides such a seasonal pattern in breeding? Why seasonal pattern breeding? For example, she was saying why light and other abiotic factors influencing endocrine pathway, especially when you said some cats were, started cycling after seeing a light. Is it conserved, such a plasticity, she was asking?
Yeah, I'm really sorry. It's a little bit hard for me to understand the words. I'm just kind of looking through the chat to see if this is a question that's been written down.
This is the question answer question.
Okay. So they want to know, say it again. I'm going to read your lips.
What decides such a seasonal pattern in breeding?
What is something pattern in breeding? Some kind of pattern?
Yeah. And she was saying how abiotic factors influencing.
So the factors that influence breeding.
Abiotic factors.
What kind of factors?
Light, other than biological factors.
Okay. Oh, so like what triggers seasonal reproduction?
Yes, especially you are saying there is a some lights, artificial light made them to breed. She was asked a question, how this happened? Is there anything animal has plasticity? Is there any phenomenon it has its own? If the light increases, it changes. That's true, but she was asking what makes. Yeah.
Yeah, well, okay.
This is the first question in the QA. You can read it in the QA, the first question in the QA.
I think I'm finally getting it. Yeah, I mean, what are the triggers for starting and stopping reproduction? And I think, let me scroll all the way to the bottom, but I think, seasonal patterns, would fluctuate. So yeah, I think that there is some plasticity to it. It's really very species specific. And I'm just sort of thinking back on the work that I did with all those different cat species, and that there are clearly some felid species that are being triggered by more likely photoperiod. And they'll do experiments like with domestic cat. You can do an experiment where you keep the diet the same, and then you manipulate the photoperiod. And since their breeding season is triggered by the photoperiod, then that gives you a good idea that it's probably not a nutritional thing. But I think there are other species that are probably triggered more by, it's still environmental, but it's triggered more by nutrition. So if you look at, elephants, for example, that, start going into must, the males start getting into breeding shape when the green grasses are growing and they're in a much higher plane of nutrition. So, and so there are some cats that seem to be primarily photoperiod driven, but there are also some desert cats that don't follow the seasonality as much as they do the nutritional, factors that are in the environment. So, it's one of these things. I think people are still trying to understand what are the main drivers of it. But I would, if I had to guess, I would say, and a lot of times you can't tease it apart, because when you have an environmental change, it's also associated with a nutritional change. But those are the two factors that are probably the most important when it comes to stimulating seasonal breeding.
There's a question, what does hormone lag time mean?
Oh, yeah. Okay. So hormone lag time means that when the hormone is in the blood and it's going to be excreted in the urine or the feces, it takes a while for that hormone in the blood to actually leave the body. So the gut transit time, for example, is what determines when a hormone that's in the blood actually gets excreted into the feces. So if you were to feed an animal a non-digestible color marker, what you would do is you would wait and see how long that color marker comes out in the excreta. And that's basically your lag time. So I think I probably forgot to say that knowing the lag time is really important, because if you want to say, I want to know what impact, I've put these two animals together for breeding and I want to know whether that stimulated their cortisol or not. Is it stressful to put these animals together for breeding? Well, you wouldn't collect the fecal sample on that day, because the hormone would not have gotten in the feces yet. You have to wait for that hormone to go all the way through the body and then come out in the feces. So we very often build in lag times in our experiments, where we would have some kind of an event, and then we don't collect the fecal sample until 2 days later, because we know the lag time in the elephant is anywhere from 2 to 3 days. So that's where that's really important, especially if you want to correlate hormones with behavior. Can I use hormones to say whether an animal is in estrus or not? It's really important to know when the lag time is, because it's that lag time that determines what sample is most appropriate to compare with your behavior.
Thank you. So there's other question, what is the reason for 2 follicular waves?
Yeah, I wish I knew. Well, I think I tried to say it a little bit, in that the waves now, the way the follicles grow and develop, are very different between the first and the second wave. And the people that have done a lot of work on that aspect of it are Thomas Hildebrandt and Imke Luters, that will follow an ovarian cycle using ultrasound. So they'll actually see the ovary over time. They'll do an ultrasound every day. And what they were finding is it looks like that first wave and that first LH surge, it doesn't ovulate anything, but those follicles that were developing become luteinized. And they just sort of are, I think, static until the next wave and the next LH surge. And then all of a sudden they become accessory CL. So they then become active in producing progesterone. And the only thing I can think of is the elephant is really big. They have a lot of blood volume. And so they would need to produce a lot of progesterone in order to maintain a pregnancy. And so I think this double LH surge makes sure that they've got adequate luteal tissue to make high enough concentrations of progesterone to maintain a pregnancy if they should get pregnant. And then there's another point that, you know, people have talked about. And there is some indication that those both waves, you know, do produce some kind of pheromones. And so I've heard reports that a bull might actually recognize the first LH surge thinking she's in estrus, but then maybe he doesn't breed her again for another 3 weeks. So there is some other hormonal or pheromonal thing going on between those two LH surges, which could potentially be a signal to other males in the area that I'm getting close to ovulating. So these bulls will start coming around a little bit earlier than if they just waited for the second LH surge. So it could be an early warning system that the female has to let bulls know that, hey, come over, I'm getting ready to ovulate.
The other question is, why some species abandon the cubs in captivity? For example, in big cats, normally sometimes they abandon, they eat up their young cubs.
Oh, they eat the infanticide?
Yeah. Why?
Yeah. I don't know how many I don't know how many species actually do that, other than the lion, of course, is very well known for that. You know, it's one of these evolutionary mechanisms to maximize the genetic, passing on the genes that the male lion has developed, because he'll come in. These male coalitions only stay with a pride for a few years, really. They turn over fairly quickly. So I think that's why they come in, and they don't want any of the other lions, cubs to survive, because they want their genes to survive. So they come in and they kill all the cubs, and then the females very quickly start to recycle, and then they'll get pregnant right away with the new coalition of males. And so then that just is a better way of ensuring that their cubs are the ones that are going to survive. So it's kind of a brutal circle of life, I guess you would say. Oh, I think, are you muted?
Someone asking, can we monitor pregnancy in giraffe and elephant?
Can you monitor what?
Pregnancy.
Pregnancy. Oh, pregnancy. Yes, very easily in both urine and feces. And in fact, in Europe, they do all of their hormone assessments using urine, and that's very effective. And you can do it with feces as well. So the progestogens come out in both urine and feces in the elephant, so you can diagnose pregnancy very easy. The pattern in the elephant when they're first getting pregnant is really interesting, in that the progesterone kind of goes up, and then it looks like it's going to come down like a normal luteal phase, because they cycle, it goes up, it starts to come down, it gets about halfway down, and then it goes up a little bit higher. And so it's very easy to kind of see where that switch is taking place, I think, is maybe the accessory CLs kick in, and then implantation takes place. But yeah, and giraffe are interesting because they have a really short estrus cycle. They cycle about every 2 weeks, but they're very clear cycles using progestogens, either urine or feces or blood. And their pregnancies are also very easy to see.
There's another question. Is there any difference between captive and wild animal in hormone cycling?
Well, I would say that what we know about, I think if there's a difference in cycling between wild and captive, it has to do with whether the captive elephant or whether the captive animals are being housed or managed in a way that kind of optimizes their welfare. So from what we've learned of African elephants is they cycle just fine in the wild, but if conditions aren't appropriate or optimal for them in captivity, that does suppress their cycles. So in that regard, if it's, yeah, I think there can be differences, but it's very species specific. You know, some animals cycle beautifully no matter where they live. Others, you know, have more problems.
There's another question. Can we predict the musts from fecal samples? Fecal hormone.
Predict must in the bull elephants? Yeah. You can't predict must, but you can monitor must. You basically the testosterone starts going up and then they start showing must signs. So it is a way of knowing for sure that a bull is in true must, but it's really more predicted by their annual cycle. You know, certain bulls, you know, will, you know, come in at certain months of the year. So, but you can monitor must with testosterone.
Okay. I think we have answered all the questions raised in this forum. I asked my colleague to find out, is there anything YouTube available? Is there anything YouTube message? Questions?
Manu? No, there aren't.
No question.
No question. Good.
There's one more question. Is the fecal cortisol value increases only negative environmental conditions or also positive environmental conditions?
Oh, yes. So cortisol is kind of like a metabolic hormone. So it's not just increased, you know, when there's negative things going on. It can also be increased during positive things. And so that's what makes it very tricky to interpret. So it can go up in positive environmental conditions. I mean, if they, you know, if let's say an animal is traversing through the desert and they find a water hole and it gives them a chance to swim and splash and that sort of thing, I would bet money that those cortisol levels were probably going up, and that would be a very positive thing. So yeah, that's what makes, so what we always say is when you're using cortisol as a proxy for stress, you really need to look at other things at the same time. So you need to correlate it with their behavior or their health or, you know, whatever else is going on in their life in order to properly interpret it. But yeah, and the cortisol in some species like elephants, for example, it goes up very regularly during the follicular phase of the cycle. So it's actually a cyclic hormone in female elephants and potentially other females as well. And that's just a normal fluctuation.
Yeah, thank you. Thank you so much for your questions, patiently answering all the questions. I may ask my colleague Manu to actually say thanks once again. Yeah.
Yeah. Thank you, everyone. A small note to participants. Kindly fill the feedback form using the link provided in the chat box at the end of the conference on 13th November to receive e-certificates through email. And thank you everyone who joined us today. And we will end today's session now. And we will start the next session tomorrow morning at 9 a.m. IST. Bye.
Thank you, Baron, once again. Thank you so much.
Thank you very much, everyone. Thanks for listening.
Thank you. Yeah, thank you so much for your time.